So, the next part, we went through, and you saw that your exam covered up through carbohydrates, right? So there were a couple questions on there about starch and carbohydrates and why cellulose is one way and the others are a different way, okay? But what we said about macromolecules such as starch and carbohydrates, right? They are composed of what? How are they built? How are these macromolecules built? And crickets. How are they built? I don't just get starch, which is a long chain of amino acids. I don't just get it like that, right? All right, so we have dehydration synthesis by joining those subunits, all right? And that's how all these macromolecules are going to be composed, okay? So I'm going to build carbohydrates by joining monomers, by joining those subunits. When we move up now, we did that for all carbohydrates, talked about the subunits, our monosaccharides. We build them together through dehydration synthesis. Now we're going to do the same thing for lipids. Okay? Some lipids do not form true polymers. Okay? So when we talk about starch, it is glucose, 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 glucose all together, right? Long chain. That is a polymer. We're joining together the same monomers. Okay? Lipids do not form true polymers in that it's not one subunit linked to another subunit. It is one part that's linked to two other parts. They're still linked together, it's just not in succession. Okay? We're still building blocks that we're putting together, but we're just going to put them together with other different building blocks. Okay? The unifying feature of lipids is that they are hydrophobic, or they have a hydrophobic nature. Okay? So oil and water do not mix. Lipids consist of fats, okay, oils, and we'll also talk about steroids. Okay? These are mostly composed of hydrocarbons. Hydrocarbon, hydrogen, oxygen, and carbon. Right? So if I say hydrocarbon, that means they have those things. All right? And they form nonpolar covalent bonds. Right? Nonpolar covalent bonds means what? Same electronegativity, therefore the electrons are shared how? Equally. Okay? So if they're equal sharing, that the polarity of water doesn't work with that. Right? So it doesn't, so they don't interact. Okay? So we're going to talk about fats and oils, right? Lipids and phospholipids and steroids. So fats. Fats are constructed from two types of smaller molecules. A 3-carbon sugar, right, glycerol, and any, what we call a fatty acid side chain, right, fatty acids. Fatty acids is just any number length of carbon chain with a C double bond OOH at the end. That's the carboxylic acid side group. Okay? So let's draw. Glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon. All right, so let's go and put that. So here's your glycerol molecule. All right, hydroxyl attached to each carbon. All right, that's basically the important part of glycerol. It's an alcohol, okay, but it is a 3-carbon molecule. Okay? Your fatty acids, and I'm not going to draw, there are a bunch of carbons, right, in a chain, and at the end you have a C double bond O, OH. That's your carboxylic acid group, which makes that a fatty acid. Okay? So what's going to happen to join this fatty acid to the glycerol? how are we going to join the glycerol to the fatty acid? Sam? Dehydration synthesis. Okay, so dehydration synthesis, water's going to come out, and we're going to link these two things together. All right, so there's glycerol. Okay, here's glycerol on your right, and there's your fatty acids. So where's that OH going to come from? Where's the H2O going to come from?
The OH on
Right? Or vice versa, it doesn't matter. As long as you have two hydrogens and a water, right? We could have an OH from this one, a hydrogen from that one. It doesn't matter where it comes from. What point of it is the water's going to come out, and then this is going to be linked to that carbon. All right? And we're going to do this in one to each carbon. 1, 2, 3. 3. Right? Once you form these three dehydration synthesis reactions, you end up with a glycerol molecule with 3 fatty acids, also known as a triglyceride. Have you heard that before? Triglycerides. It's one of the important parts when you're asking someone has possible heart disease. They look at the amount of triglycerides or fat in the blood. Okay? So triglycerides are fats. All right? What do you notice about this chain of fatty acids? Hydrocarbons, hydrogen and carbon. Lots of them, right? What do you notice about those carbons? They're not in a straight line. They're kind of bent, and that's why I draw them like this. All right? What else? They're stable. All the carbons have 4 bonds. They are stable. What else? Anything else? What about the bonds between carbons? It's a single bond between carbons. And overall, basically what you end up with is a pretty straight line, right, with single bonds. If I was to introduce a double bond right here, okay, between the carbons. Well, actually, let's do that in a different color so you can see it. A double bond right there, right? So now we have 2 bonds between carbons. Everybody see that? Where that is? Yes? Okay. What's going to happen? Correct.
Would it break the chain?
It wouldn't break the chain. What happens to hydrogens?
You're gonna lose.
You're going to lose how many hydrogens? 2. Okay? We're going to lose 2. These 2 hydrogens are going to come off. This one leaves, and so does that one. Okay? So now I introduce a double bond. How many hydrogens are for each carbon in where there are only single bonds? There's at least 2 for each, right? Yes? Is that all the hydrogens I can fit in there? I can't fit any more, right? The carbons that make up that fatty acid can't contain any more hydrogens in this, say, in chain 1. Can they fit any more hydrogens in there? Are there any empty carbons that don't have hydrogens? No. Okay? So as much hydrogens I can fit in there is there. That is a saturated fat. It is saturated with hydrogens. Okay? But now when I introduce the double bond, these 2 are gone. Those 2 hydrogens are gone. So in the fatty acid chain 2 there, are there all the hydrogens I could possibly fit on that chain? Are they present? No. I could fit 2 more, right? So that's not a saturated fat. That's an unsaturated fat. And that's the difference between a saturated fat, all hydrogens, all single bonds, and an unsaturated fat. At least 1 double bond, and therefore it doesn't have as many, or it doesn't have all the hydrogens that could be present, or could be held by the number of carbons there. When you end up with a unsaturated fat, something happens too. This ends up bending. So the chain ends up bending a little bit, okay? And thereby forcing this to not pack together, okay? And we're going to talk about the differences between saturated fats and unsaturated fats. So there's your dehydration synthesis. That's palmitic acid, right? HOH, right, comes off. Water comes off and forms an ester linkage. An ester linkage is between your C double bond O, O, C. Okay, there's an oxygen between two carbons. And then you end up with a triglyceride. Okay, what do you notice about the triglyceride? What type of fat is this? It's a saturated fat. Does it matter? What else do you notice about the fatty acid side chains? The fatty acids that are up there. They're different lengths. It doesn't matter what the lengths are. Okay? They could be different. And that's what gives different properties of the certain saturated fats or fats in general. Okay? So you end up with a triglyceride or a triacylglycerol. Right? Those are basically the same things. Okay? And because of those long fatty acid chains that have covalent bonds between carbons that are nonpolar, that's what gives fats their hydrophobic nature. Okay? So they do not interact with water because of that. Ouch. All right? Questions on triacylglycerol? So you end up with fatty acids. They vary in length, right? Number of carbons, and they also differ in the number of bonds between carbons, which is saturated versus unsaturated. Most saturated fats. So it says here, the maximum number of hydrogen atoms, right? The maximum number of hydrogen atoms are present and no double bonds, and unsaturated have one or more double bonds, and therefore they do not have the maximum number of hydrogens present in the fatty acid chain. Questions on saturated fatty acids? All right? So saturated fats are glycerol molecules that are linked to saturated fatty acids. Okay. Oh, no. No. So here's a saturated fat. Most saturated fats are animal in origin. Okay? Come from meats, red meat, mainly red meat. Okay? Milk, butter. All right? They also they're usually solid at room temperature. And this is why. The structure of the fatty acid, okay? They're in straight lines, okay? So if all the fatty acids are in straight lines, because they are saturated, it is easy to stack them together and push them into a closed space. All right? So at room temperature, they are all stacked together and pushed into a closed space and become a solid. When you have an unsaturated fatty acid, the double bond forms a bend in this fatty acid chain. Does that make it easier or harder for them to stack things together? Harder. Makes it harder. Therefore, they don't stack together as well. Therefore, it's not a solid at room temperature. Okay? So structure, function, relationship. Most unsaturated fats are oils. They are liquid at room temperature, and they are mainly plant-derived. Okay? In some cases, you will talk about trans fats right? And you actually also have, how many of you like Oreos? Yeah, okay. Love me some double stuffs, okay? The cream part of Oreos are the best part, right? Yeah. But it's actually, if you read the ingredient label of Oreos, it says hydrogenated soybean oil. That's what it's composed of. Hydrogenated soybean oil. So soybean oil, right? Plant derived, yes? Would you expect a soybean oil to be unsaturated or a saturated fat? Unsaturated. But now I put that hydrogenated in front of it. What does that mean?
It solidifies it and turns it into saturated fat.
It solidifies it and actually turns it into, you're hydrogenated is you're adding hydrogens in here to break and get rid of these double bonds. So you take the soybean oil, which was good, did something to it, and made it not so good. But it tastes good, right? So when I say hydrogenated, we're putting hydrogens in because we can add them in there and break those double bonds. Okay? Okay, so fats made from saturated fatty acids are saturated fats. Fats made from unsaturated fatty acids are unsaturated fats. Okay, and then plant fats and mainly fish fats. All right, so omega-3s, you heard of those, right, from salmon and the lovely fish we consume. They are usually unsaturated. The major function of fats, what they do. They are energy storage. Okay? Your body can break down fats and produce energy. Okay? Your body can also take excess sugar and store it as fat. Okay? So fat's a great way for animals to carry their energy stores. Right about now, I mean, my wife used to be a bear keeper in Florida. Okay? So she took care of bears. Right? Big ones, like 1,200-pound grizzly bears. Pretty cool, right? But right about now in the northern hemisphere, what are bears preparing to do? Hibernate. And why are they doing it? What are they doing for hibernation? They're storing fat. Okay? That's all they're doing, right? They're consuming between 20 and 50,000 calories a day, right? In order to produce fat to be survive over the winter. And over the winter, they'll break down that fat, give them energy while they hibernate. That's recording. Okay. Phospholipids. Basically what we just talked about in terms of saturated triglycerides, okay? Glycerol molecule. I'm going to have that 3 carbon. I'm going to add a fatty acid to each of them. Then we're going to take one of them off, one of those fatty acid chains, and replace it with a phosphate. Okay? That, in a nutshell, is a phospholipid. Okay? So therefore you have something that looks like this. You have carbon, 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 fatty acid chain, right? Yes, you agree? Good, I hope so. But then on this last one, instead of the fatty acid chain, we're going to add a phosphate group. Okay? Easy peasy? Yes? But that phosphate group is important because what do we know about the fatty acids? The fatty acids are what? Hydrophobic. Okay, they're nonpolar and hydrophobic. So let's put that over here. They are nonpolar. Polar. Where am I from? Boston. Okay, nonpolar and hydrophobic. Okay? However, the phosphate over here, okay, that is charge. This is polar on this side. Okay? And if it's polar, what do you think it is? Will it react well with water? Yes, it will. That's hydrophilic. Hydrophilic. Man, my handwriting's getting better. Okay? So the whole phospholipid molecule, we'll say, has a hydrophilic head and hydrophobic tails. So if I was to simplify this thing down to draw it out, it would look something like this. Right? And we put a little plus up there, right? Because that's the polar end. Okay? Cool beans? Clear as mud? Awesome. So this is what it looks like. Here's your glycerol. Glycerol molecule 1, 2, 3. Two of these glycerol carbons have fatty acids. In this case, what type of fatty acid is this? Unsaturated. Okay? And there's your phosphate molecule. And we really just worry about this phosphate. We don't really think about this choline, right? There can be extensions of that off of carbons off of the phosphate, but we're really concerned about the phosphate because that is what gives it its polar molecule. So we have glycerol, the phosphate, and the fatty acids, okay? And then you end up with the structure where we look at, this is your phospholipids, your hydrophilic head and your hydrophobic tails. Now, if you had some phospholipids in there and you mix them with water, those tails would orientate themselves towards each other, and the polar head would orientate towards water, and you end up with this lovely thing right here, your phospholipid bilayer. Okay? Your phospholipid bilayer, here's the polar part, right? And here is the nonpolar. And if it's nonpolar, oh boy, that means it is what? Hydrophobic, right? And up here is hydrophilic. Okay? This is what constitutes what? The cell membrane. Okay? The cell membrane is constituted by mainly a phospholipid bilayer. Okay? And then we're going to add some other stuff to it when we talk about the cell membrane. We're going to add some proteins and some sugars on lipids and sugars on proteins and a bunch of other stuff, and you end up with a functional cell membrane. Okay? So, phospholipids, and this is important, when they're added to water, they self-assemble into a bilayer. Self-assembly of phospholipids is probably where cells originated from. Okay? During early Earth, we had chemical reactions ongoing, and at some point, phospholipids were produced and then self-assembled into membranes. All right? This gives you your bilayer of cell membranes. Questions on phospholipids? Now, both phospholipids and triglycerides, or fats, they are composed through dehydration synthesis. They're going to add subunits together to form polymers, or pseudopolymers. The last lipid we're going to talk about does not form by dehydration synthesis. Okay? It actually starts as a backbone from cholesterol, and then they're altered. Okay? Steroids are your last lipid. All right? They have a carbon skeleton consisting of 4 fused rings. When I was taught this, we were told it has 3 rooms and a bath. Okay? 3 6-member rings. Whoops. Let's minimize that first. Okay? 3 hexagonal rings. 1. that are fused together, 2, 3, and then a little 5 ring. That's your basic structure of cholesterol. 4 fused rings. Okay? And then off of these carbons, because this carbon right here, still I only put in 2. Right? All of these vertices within the ring is a carbon, and each of them can have up to 4 bonds. So I can have hydrogens there, I could have different side groups, which end up giving this functionality. Okay? But it all comes from cholesterol. Okay? Cholesterol is also an integral part of cell membranes. Okay? That picture I showed you a little bit ago of the phospholipid bilayer, what did you notice about what was not there? No proteins, no cholesterol, and there were lots of phospholipids in a small space. Yes? Yes or no? Yes. Cholesterol, its function in cell membranes is that it kind of wedges in between those phospholipid tails and prevents them from packing in too much. If the tails pack in too much, the membrane becomes rigid and can break. So cholesterol prevents that from happening. All right? Here's cholesterol. That's the structure of it. So here's your, 1, 2, 3, 4. Right? And all those other groups are side chains. So now you start with this substrate material, right? Raw material cholesterol. Move the side chain, change the groups around, maybe change the bonds around in here, arrange it, and then you end up with steroids. What do you know steroids? What are they?
Anabolic agents.
They're anabolic agents. Give me examples of steroids. Testosterone. What else? The opposite of testosterone. Estrogen. Progesterone. Thyroid hormone's a steroid. Okay? So, examples of steroids. Okay? Estrogen and testosterone are the same structure. They have the 4 fused rings, but they have the side groups that end up determining what they do. Okay? And how they work. Questions on lipids? So let's review lipids first. Lipids, 3 types. What ones we talked about? Go. Phospholipids, steroids, and fats. Fats are composed of what? What are the subunits of fats? Glycerol and fatty acids. Okay? And we talked about 2 types of fats, saturated versus unsaturated. Which one has a double bond? Unsaturated. Okay. Phospholipids. What's that subunit? 2 fatty acids. What else? A glycerol and a phosphate. Okay? And then we have steroids. They don't have any subunits, but the basis of steroids are cholesterol. All right? They come off the cholesterol backbone. Examples: estrogen, testosterone, progesterone, thyroid hormone. Okay? Our next macromolecule, and write this down in your book, the most diverse in terms of function. Okay? The most diverse in terms of function. We know what fats are used for energy. Carbohydrates are mainly used for energy or structure. Okay? Steroids are used for membrane fluidity and anabolic agents, right? And then hormone responses. Okay? Proteins do all of those things. They're used for structure. They do enzymes. They are used for hormones, receptors. They are part of the cell membrane, right? They are the most diverse functionality, right? Functional-wise, right? They also include defense, antibodies, transport, cellular communication in terms of hormones, movement, right? Contraction and structural support. And they're so much that they account for about 50% of the dry mass of the cell. All right, so here's some examples. You have enzymatic proteins, defensive, storage, transport. All right, you should know the basic list. Okay, if I asked you to provide a list of functions that proteins can do, there's about 6 or 8 of them. Okay, you should be able to know that. All right, hormones, receptors, structural, contractile, motor proteins. The point of this is so that you understand the diversity in function that proteins have. Okay? There's an ongoing debate among molecular biologists that really it's like an argument. You know, what comes first, the chicken or the egg, right? Or what's more important, the chicken or the egg? What's more important, proteins or DNA? What do you think? Well, no one agrees on it anyway. You ask a protein biologist, he'll say proteins. They do all the work of the cell. You can't do anything, and the cell can't do anything in the cell, and the cell can't reproduce without proteins. Okay, fine. You ask a geneticist or molecular biologist in terms that studies DNA, and he'll say DNA, because it controls what the proteins do, right? It controls how proteins are function and what they... Double-edged sword. It doesn't really matter. They're both very important. However, proteins do the work, okay? They do the work, all right? And one of the important types of proteins are enzymes. Next week in lab, you're going to be doing enzymes. Okay, enzymes function as biological catalysts. The reactions we talk about, the reactions that happen in your body, they could happen outside of your body at the same temperature, right? But sometimes it takes a little while. So you would agree that your body's mainly water, right? Okay, so if I had a beaker of water up here, all right, and I put enough sugar in that water that is in the same amount that you have in, I don't know, iced coffee. Okay, how many of you like sugar in your coffee? Okay, so we throw that in there. What's going to happen? Eventually that sugar, sucrose, will break down into glucose and fructose. Yes? You agree with that? How long is that going to take sitting here on the desk? Well, I'll even heat it up a bit. I'll heat it up to 98.6 degrees Fahrenheit. How long is it going to take? Pretty long time, right? What if I did starch, cornstarch? Same thing. Sat it up here and let it sit. It's going to take a while, right? Okay. Now I add an enzyme. I have purified salivary amylase that I put in that starch solution. Now what happens? It speeds up the reaction. Okay? That's what's happening in your body. Okay? Without the presence of enzymes, reactions would take too long. And we need it to happen quickly. Okay? So catalysts will speed up reactions, make them go faster or at a lower temperature or at a permissive temperature or permissive pH, whatever it might be, right? But what are they doing? They're speeding up reactions. They're allowing reactions to occur that maybe wouldn't occur as quickly as that given if the enzyme wasn't present. Okay? All enzymes are proteins, but not all proteins are enzymes. True or false? True. Excellent. Okay? All proteins are peptides. True or false? all proteins are peptides. True or false?
True.
Would they be polypeptides?
They could be. The best answer would be true, because no matter what, a protein, and this gets to the definition and the distinction between a peptide, a polypeptide, and a protein. When I say the word protein, I'm assuming that is the functional structure of that protein. Okay? Okay. Now, a peptide is a linkage between amino acids. All right? And all proteins are chains of amino acids that are linked together. So yes, by definition, all proteins are peptides. Usually, they are also polypeptides, more than a certain 20 amino acids or so. Okay? But the definition of protein assumes functionality. Okay? Means that is how that protein works, or that peptide works. It is in its working structure. Okay? So, I did mention that proteins are composed of amino acids. Okay? There are 20 naturally occurring different amino acids. All right? All of which have the same structure, basic structure. Okay? They have a carboxyl group and an amino group. What's a carboxyl group? It's a functional group composed of what? Here's your carboxyl. C, double bond O, OH. Carboxylic acid. Okay? And on the other end, an amine group is a nitrogen with 2 hydrogens. Okay? In the middle of these two is a lonely carbon, or this, what we call the central carbon. Okay? All amino acids share this structure. Okay? There's a little hydrogen here. All amino acids share this structure. How many bonds does that central carbon have right now?
2, 3.
That's 3. Okay? The last one, the last bond, is what gives each amino acid its different structure or its different property. This is what we call, let's do it in purple, the R group. Okay? The R group. It could be as simple as CH3. Could be as simple as a hydrogen. Okay? It could be as complex as this ring structure with some double bonds, maybe whatever else, sulfur groups, whatever it might be. Okay? But the difference between a proline, which has a certain R group, okay, and a lysine is just this. It's just the R group itself. The R groups change, you have a different amino acid. So there are 20 different amino acids, there's 20 different R groups. Okay? There's your central carbon, right? And that's where the R group is. You change it, you change the amino acid. Here are your, let's see, I think that's 20 amino acids there. Glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, proline, serine, threonine, cysteine, tyrosine, asparagine, glutamine, and then you have the aspartic acid, glutamic acid, lysine, arginine, and histidine. Okay? They are, now, the properties of each amino acid, they're grouped differently. We have nonpolar, polar, acidic, and basic. What's giving them those different properties? What's giving these amino acids those properties? The R groups. Okay? Because the R groups are nonpolar, that group is nonpolar. All right? There's nothing changes about the basic structure of these, just the R groups. Okay? And the R groups are important when we start talking about structuring these proteins. Okay? Questions on amino acids? The structure of amino acids. All right? So polypeptides. Now we're going to build a protein and a polypeptide. We're going to use amino acids as our subunits. And what are we going to do to build polypeptides? What are we going to do? What does a cell need to do to join amino acids together? I've beat this dead horse already, so you guys should know. What is it? Dehydration synthesis. Okay, good. All right. So when we synthesize a new peptide, we're linking amino acids by a bond, a peptide bond. We're linking one N group, N terminus, or the amino end of one amino acid to the carboxy end of the preceding one. Okay? So here, let's say this is a central carbon. Here's your C double bond O, OH of one amino acid. And now I'm going to have the NH2 of this amino acid. There's your central carbon. There's an R group. There's another C double bond O, OH. Right? Yes? Agreed? Okay, good. Now, this water is going to come off, and then we're going to make this bond between carbon and nitrogen. That bond between carbon and nitrogen is your peptide bond. Okay? And so you have amino acids 1, amino acid 2, and we could add amino acid 3 down the line, and so on. Right? Each time having a successive dehydration synthesis reaction. Questions on peptide bond? All right? So polypeptides then are chains of amino acids with lots of peptide bonds. Right? They could range in length from a few, 3, 4, 5, to more than 1,000. The largest protein out there is protein called titin. Right? It has over 30,000 amino acids that are linked together. Okay? There's the diagram, right? Peptide bond, dehydration synthesis. We're adding one to the next. All right? And there, how do we know the chain of amino acids? How do I know it's going to be that one, and this one's coming next? How do I know that's the way it's going to go? What determines the sequence of amino acids? Well, it's really DNA, which RNA is carrying the message.
The nucleotides?
Right, the nucleotides that are there in the DNA, right? So DNA is going to be transcribed by RNA, and the nucleotides that are set up. Yes. So the first structural importance of proteins is the sequence of amino acids. The amino acid, it's going to be methionine, proline, lysine, instead of methionine, lysine, tryptophan. Okay? That's two different proteins, and that is important structurally. Okay? So we have structure. Right? There are 4 levels of protein structure that we need to know about. Okay? And a functional protein, what we said about definition, protein assumes functionality. Okay? A functional protein has unique structure, important structure. Structure and function relationship again. Okay? So, first level of protein structure, right, primary, is the sequence of amino acids. Okay? From there, the sequence of amino acids folds and twists, really twists and turns into secondary structure. Secondary structure. We have twisting and turning, right, to form what we call alpha helices and alpha helix, or a beta sheet. Okay? And that has to do with hydrogen bonding between amino acids that gives the amino acid a stable structure. All right? Mostly all proteins have at least what we call tertiary structure. Right? And this is globular folding. Now that twisted sequence of amino acids in turn kind of folds upon itself and forms a globular structure. In terms of one polypeptide, that is the last step. Okay? So if I have one chain of amino acids, amino acid 1 through 25, it will fold a certain way and then it will stop there. That may or may not be the functional part of that protein. Because we have one more step of protein structure, and that's quaternary structure. Okay? Okay? The fourth degree, or the fourth step, is that multiple polypeptides come together to form a functional protein. To form a protein. Okay? Best example, best known example is hemoglobin. Hemoglobin is 4 separate subunits, 2 alpha globin proteins and 2 beta globin proteins that come together and form one functional protein. Okay? But they have to interact all together to do that. Okay? And you end up with something that looks like this, ribbon models and groove models to show you what structure is important for. Okay? And your structure determines what? Okay? Determines function. Changing that structure will change function. We'll talk more about this on Monday. I will get your test grades up either this afternoon or sometime tomorrow. Okay?